Imagine an organism that can, like, survive being boiled in literal battery acid, or frozen completely solid inside an Antarctic glacier.
Right, or even just thrown unprotected into the irradiated vacuum of space.
Exactly. You expose it to solar radiation that absolutely shatters its DNA into hundreds of little pieces. You bring it back inside, you add a single drop of water, and it doesn't just wake up. I mean, it lays healthy eggs.
It's unbelievable, and for decades we've just operated under this assumption that life is, well, fragile.
Yeah, like it's this delicate accident that needs a pristine, perfectly balanced little cradle to survive.
Yeah.
But today, we are completely tearing up that biological rulebook.
We really are. We're completely dismantling that whole anthropocentric lens. Because for generations, biology textbooks and, frankly, our entire space exploration strategy, they were constrained by our own frailties.
Right, because humans need a very strict, gentle equilibrium.
Exactly. We need a narrow temperature band, we need heavy atmospheric shielding from cosmic rays, and we need a lot of neutral liquid water.
Otherwise, we just die.
Right. So the prevailing logic was simple, but flawed. We assumed if an environment would coagulate human proteins or, strip our cellular membranes or crush our bones, it was categorically just a dead zone.
We were viewing the entire universe through the lens of our own limitations. So, welcome back to Stellar Lab. It is so good to have you here with us today.
It's great to be back.
Today's mission is an exploration into the absolute fringes of life. We are pulling from an extraordinary body of research about extremophiles. Specifically, we're looking at the findings from that amazing book, Grenzenlos Resilience or Limitless Resilience.
Such a fantastic deep dive into this topic.
It really is. So we are venturing into environments that don't just push the boundaries of habitability, they just obliterate them entirely. We're talking about organisms that treat a planetary blast furnace or a bath of corrosive chemicals, not as a hazard, but as home.
And I think the distinction between surviving and thriving is so critical here.
Oh, absolutely.
These aren't organisms just hanging on by a thread, secretly wishing they were in a nice temperate pond.
Right. They're not just tolerating it.
No, their entire biochemical architecture is fundamentally engineered for extremes that would instantly denature standard Earth life.
Which is just wild to think about.
It is. And by examining the mechanisms they use to master these environments, we aren't just adding fun little footnotes to an encyclopedia. We are expanding the whole horizon of astrobiology.
We're radically altering the mathematical models for where life can even take hold in the cosmos. So let's start by removing the single most fundamental pillar of terrestrial biology.
The sun.
Yep. Every food chain we track at our standard ecosystems, it always traces back to this single, supposedly non-negotiable energy source.
Right. Photosynthesis drives the plant life, the plants feed the herbivores, and they feed the apex predators.
Exactly. You take away the sun, the surface freezes, the food web collapses, and the planet just dies. But if we descend into the abyssal zones of our own oceans, specifically along the mid-ocean ridges, we find ecosystems that have never seen a single photon of sunlight.
Never. We're looking at depths where tectonic plates are literally tearing apart. They're exposing raw magma to the ocean floor.
Which just sounds like a supervillain layer.
It totally does. And the physical parameters of these hydrothermal vents are just staggering. I mean, the hydrostatic pressure down there is immense.
Like, how immense are we talking?
It's enough to crumple a modern nuclear submarine like a piece of tinfoil.
Wow.
Yeah. And because you have raw magma interacting with the ocean, the water is superheated to well over 300 degrees Celsius.
Okay, wait, I need to pause you there because at sea level, water boils and turns into steam at 100 degrees Celsius.
Right, exactly.
So sitting at over 300 degrees, this isn't just boiling water.
No, it's not boiling. The crushing weight of the entire ocean above it physically prevents the water molecules from expanding into a gas.
That is insane.
It creates what we call a supercritical fluid. It's a liquid, but it has the kinetic energy of a gas. It's hot enough to instantly melt lead.
And yet, clustered right around these vents, we find these dense, thriving communities of hyperthermophiles.
Which is just mind-blowing. These microorganisms have completely decoupled their existence from solar energy.
Right, in the pitch black.
Yeah. In total darkness, they rely on a process called chemosynthesis.
Okay, so how does that work compared to photosynthesis?
Well, instead of capturing photons from the sun to convert carbon dioxide and water into energy, they capture chemical energy directly from the toxic minerals that are spewing out of the Earth's crust.
So they're using, like, hydrogen sulfide.
Exactly. Hydrogen sulfide is a highly toxic, corrosive gas. I mean, it inhibits cellular respiration in most animals, but for them, it's their primary metabolic fuel.
It's a complete inversion of what we consider a life-sustaining resource. It's honestly like discovering a bustling metropolis where millions of people are just running their homes and their cars entirely on toxic smog.
That's a perfect way to put it.
They're just breathing in the smog, metabolizing it, and building complex biological structures out of it.
And the biochemical adaptations required to actually pull this off are phenomenal. Because at 300 degrees Celsius, normal proteins just unfold.
Right, they basically cook.
Yeah, the hydrogen bonds that give proteins their specific 3D shapes just vibrate apart from all that kinetic energy.
So how do the hyperthermophiles hold themselves together?
They solve this by utilizing highly stable enzymes. These are often held together by much stronger covalent bonds, like disulfide bridges.
Oh, okay.
It essentially locks the protein structures in place even under extreme kinetic stress. They've reinforced their internal molecular architecture with heavy-duty biological rivets, basically.
Biological rivets, I love that. And think about the astrobiological implications of that for you listening. If you could somehow magically extinguish the sun tomorrow, the Earth's surface would just freeze solid.
Oh, instantly.
The entire surface biosphere would be totally eradicated in a matter of weeks. But these abyssal ecosystems, they wouldn't even notice.
They really wouldn't. They would just keep multiplying. Their energy economy is completely closed loop with the planet's internal geothermal heat.
That is so cool.
It proves that a planet could be entirely dark. I mean, it could be a rogue planet.
Like a planet ejected from its star system?
Exactly. Just wandering through the freezing void of interstellar space with a surface temperature near absolute zero.
Wow. But as long as it has that internal heat.
Right. As long as it retains internal radiogenic or tidal heat and has liquid water beneath some global ice sheet, it could harbor a biosphere just as complex as the ones we find at our mid-ocean ridges.
Okay, so sunlight is definitely not a requirement. And extreme heat and pressure are basically just engineering challenges that biology has clearly solved.
Precisely.
But if we swing the pendulum in the exact opposite direction, like taking away the heat entirely, we hit a very different kind of physical barrier.
We certainly do.
So let's leave the boiling ocean floor and move to the perpetual deep freeze of the Antarctic ice sheets, home to the psychrophiles.
Right, and the assumption with extreme cold has always just been dormancy.
Like hibernation.
Yeah, we just imagine organisms locked in this state of suspended animation, frozen solid, just waiting out the centuries for a thaw.
But that's not what's happening here, is it?
Not at all. Psychrophiles are actively metabolizing, they're reproducing, and they are navigating through solid glacial ice at temperatures 20 or 30 degrees below zero.
See, navigating through solid ice sounds like a physical impossibility to me.
It does, right, but it really comes down to the microscopic architecture of frozen seawater.
Okay, walk me through that.
When ocean water freezes, the crystalline lattice of the ice is actually remarkably pure. It actively expels the salt ions as it forms.
Okay, so you get pure freshwater ice.
Exactly, a matrix of solid freshwater ice. But woven all throughout that matrix are these microscopic, highly concentrated veins of liquid brine.
Oh, because the salt was expelled into those veins?
You got it. And because the salinity in those veins is just so incredibly high, the freezing point of that water is suppressed dramatically. It remains liquid far below zero.
So the psychrophiles are actually inhabiting this microscopic, super chilled liquid network inside the ice.
That's exactly where they live.
Okay, but remaining in a super chilled liquid is only half the problem, right? I mean, if I put regular organic tissue in a freezer, the water inside the cells crystallizes.
Right, and that's fatal.
Yeah, those ice crystals act like microscopic daggers. They just pierce and rupture the cellular membranes from the inside out.
It destroys the cellular integrity completely.
So how on earth are psychrophiles avoiding this intracellular crystal formation?
They deploy something called antifreeze proteins.
Antifreeze, like in a car.
Kind of, yeah. These are highly specialized molecules that the cell produces and distributes internally. The very moment a microscopic ice crystal begins to form inside the cytoplasm, these proteins physically recognize the shape of the ice lattice.
They can recognize the shape.
Yeah, and they bind directly to its surface. They basically capped the crystal.
Wow.
By binding to the growing edge of the ice, they physically block any more water molecules from joining that lattice. It halts the crystal's growth before it can become large enough to puncture the cell membrane.
So they are essentially recognizing the dagger while it's still molecularly tiny and snapping a protective sheath right over the blade.
That is a brilliant way to visualize it.
That protects the interior, sure. But what about the membrane itself? Because extreme cold causes lipid bilayers, the walls of the cell, to just stiffen and freeze solid.
Right, they lose their fluidity.
And if the membrane loses fluidity, the cell can't transport nutrients in or push waste out. It's game over.
To combat that membrane stiffening, Psychrophiles completely alter their lipid composition. They pack their cellular membranes with polyunsaturated fatty acids.
Oh, wow. That is a brilliant piece of biological engineering.
It really is.
Because polyunsaturated fats have those little chemical kinks in their hydrocarbon chains, right?
Oh.
Which means they can't pack tightly together to form a solid.
Exactly.
It's identical to the logic of winterizing a vehicle's engine oil.
Oh, that's a good comparison.
Like, if you use a high-viscosity summer oil in an Arctic blizzard, the oil turns to sludge, the engine component seize up, and the vehicle dies.
Right, you have to swap it for a thinner, more fluid synthetic oil to maintain that mechanical movement.
Exactly. So by building their membranes with kinked, polyunsaturated chains, Psychrophiles ensure their boundaries remain totally fluid and permeable even when the whole environment is trying to lock them into a solid state.
Precisely. And when we apply this to the broader solar system, the implications are just staggering.
Oh, for sure.
I mean, we've long observed worlds like Jupiter's moon Europa or Saturn's moon Enceladus.
Like, these are massive global oceans entirely encased in shells of ice that are miles thick.
Yeah. And if life on Earth has evolved to actively metabolize inside the brine channels of an Antarctic glacier using antifreeze proteins and winterized lipid bilayers, well, there is zero biological constraint preventing similar organisms from thriving within the icy crusts of those moons.
The habitat isn't just theoretically viable. We literally have a working proof of concept right here on Earth.
We really do.
Okay, so we have conquered the physical extremes of temperature and pressure. But physical parameters are really only one axis of environmental hostility.
Right. There's also chemistry to worry about.
Exactly. What happens when the environment itself is actively trying to dissolve your molecular structure?
That brings us to the acidophiles and alkalophiles.
The chemical survivalists.
Yes.
Let's delve into environments like hyperacidic volcanic lakes or these subterranean acid mine drainages where the pH hovers near zero.
Which is unimaginably acidic.
Right. An environment with a pH that low is just flooded with hydrogen ions. It is literally equivalent to swimming in battery acid.
It'll strip electrons, it'll destroy lipid membranes, and violently denature your proteins.
And you can't just build a thicker wall to keep that out, can
You
you?
really cannot. A passive defense is mathematically impossible against that kind of chemical gradient.
So what do?
Acidophiles survive through sheer, continuous, active exertion. They are equipped with these highly specialized cellular pumps embedded right in their membranes.
Good.
Because the external environment is hypersaturated with protons, those hydrogen ions, those protons are constantly leaking into the cell.
And if the internal pH drops, the organism's DNA just unravels instantly.
Instantly. So these cellular pumps are operating nonstop. They capture the intruding protons and forcefully eject them back out into the acidic environment.
Against a massive concentration gradient.
Exactly, against the gradient.
Like being in a sinking rowboat made of acid, and you were just frantically bailing out the toxic water with a tiny bucket at hyperspeed just to maintain a neutral internal environment.
That is exactly what it's like, and the thermodynamic cost of that bailing is astronomical.
I bet.
A huge percentage of an acidophile's total metabolic energy budget is dedicated solely to running those proton pumps. Their entire biochemical economy is structured around funding this continuous expulsion.
Wow. And do we see the inverse of this?
We do. We see the exact inverse mechanism in alkalophiles, which inhabit high pH environments like soda lakes.
Right, because a highly alkaline environment has a massive deficit of protons. So the environment wants to pull protons out of the cell.
So the alkalophiles just run their pumps in reverse. They aggressively scavenge the few available protons from the external environment and pump them inward.
Just fiercely guarding their internal neutrality against a highly basic exterior.
Exactly.
This whole theme of fiercely guarding internal equilibrium brings up the halophiles too, right? The salt lovers.
Oh yes, halophiles are fascinating.
They thrive in environments like the Dead Sea or these ancient subterranean brine pools. And high salt environments are fundamentally desiccants.
Right. Through the laws of osmosis, water always moves toward the higher concentration of solutes.
So if a normal cell finds itself in a heavy brine, the water inside its cytoplasm